Thank you so much for inviting me to talk to you. And today I'm really going to share with you a story that's more of a biology puzzle and some of the things we know so far about this puzzle. And I'm really hoping that I'll be able to find some more computational collaborators who are excited by this research who might want to help us piece it together. So in the United States, it's a huge clinical problem that many Americans, almost 1% of the population actually, has suffered a limb loss. And so a major limb loss would be a hand, an arm, a foot, or a leg. And this number is expected to rise because of the risk factors that may necessitate an amputation are becoming more common in the United States, such as diabetes. And in our lab, we are studying a particular kind of salamander called an axolotl. And all salamanders studied to date are actually capable of regenerating their entire limbs throughout their entire lives. And this is shown here in this video. And the video is going to loop. So what I want to draw your attention to when it loops around in a second is just the little nubbin at the beginning of the video. And so this little nubbin, which you'll see again here after the amputation, has a name, and it's called the blastema. And the blastema is really a structure that's almost like an on-demand niche that the animal creates, uses, and resolves, and it goes away. And so in this little pseudo niche are the progenitor cells that are going to be used to make that new limb. After the full regeneration, which in this case is about 8 weeks, there's no scar visible on the outside of the animal, and you can't tell which limb was amputated in the first place. So this is really an awesome process. And because salamanders have basically all of the same tissue types that human limbs have, we really think this is a blueprint for how to understand how limbs can be regenerated so that we can have hypotheses that people working on therapeutics can use later to perhaps one day stimulate it in human beings. So how does this work? Some of the steps have been appreciated at sort of the gross anatomical level for several centuries, actually. But we only really right now have the tools in hand to begin to dissect this process at a more a priori molecular level. And the main things that you need to know are that after the amputation, a very important kind of skin forms called the wound epidermis. That wound epidermis is required for regeneration. And then the next thing that happens is that these activated progenitor cells collect underneath the wound epidermis to make the blastema. And there they remain relatively undifferentiated, and then they proliferate a lot, and then they undergo differentiation and pattern formation to make the new limb. And in my lab to date, we've actually done quite a bit of work to try to understand the biology of these blastema cells once they're already made. So in collaboration with the Broad Institute, in the beginning of the lab, one of the things we did was just take this really a priori bulk sequencing approach to understanding what are the genes that are very specifically expressed in blastema cells, in the hopes that those will give us some clues about what blastema cells are doing and how they're doing it. And so here you can see in this heat map that this represents two different kinds of blastemas. One's taken from an upper arm amputation and one's taken from a hand amputation. And these are all the genes, there's about 150 of them, that are very highly expressed in those blastemas versus all of these other tissues. And among the other tissues are the presumed parent tissues, so the cartilage, the bone, the muscle, et cetera. And what we learned so far about those through CRISPR and experiments and other knockdown kinds of experiments are that largely these genes are required for proliferation, for cellular survival, and for maintenance of genomic integrity during this process. And so while we think this is interesting and these provide important clues to what blastema cells are doing, as you know, the bulk sequencing approach has its caveats. And so we really wanted to take this down to a single cell level. And so we recently completed a study whereby we sequenced the single cells in that blastema and indeed in the intact limbs as well, and time points towards generating the blastema to try to understand what are the different cell types in there. And I'm not going to get into this today because it's now published, but we made an atlas of the different stages of wound healing, blastema formation, and then a full-blown blastema shown here. And so this work was really interesting, and one of my postdocs has now started his own lab to look at the role of the immune cells that were found in this study. But we really also started realizing that if we really want to understand this process, we actually need to go a little bit earlier, so before the blastema is even made. And we also need to know more about the signals that control this process. And so right now in the lab, we're using another single cell sequencing technology that is based on this retroviral mediated cellular barcoding shown here, which works really well in the axolotl, to try to make a map of how all the cells are related to one another, and then also what are the genes that they express along these trajectories of becoming their ultimate differentiated cell types. And then finally, I would like to argue for a sort of new model in thinking about how this process works, and that is shown over here on the right. And so traditionally, we think of all the activity is happening on the site of amputation, and we'll talk about that in a second. But in this two-step model, which I would propose, is that in this mix of different cell types shown here, that actually the first thing that happens is an activation step, meaning some cells re-enter the cell cycle, shown in hot pink. And of these cells, only some of them go on to convert to blastema cells, shown in aquamarine. And this conversion event is the thing that is really quite unique to these highly regenerative super regenerator animals, because what I'll show you in a second is that other animals also undergo these activation steps, but they don't actually culminate in conversion to blastema cells. And so what this really means is that we need to understand, how do you get activated, but once you're activated, how do some cells get converted and some do not? And so right now in the lab, we are setting up to do some screen-based studies to look at this both at a cellular and a whole animal level. Okay, so one of the questions that we realized we had stumbled into was this question of, is everything going to be just local? And historically, most of the research effort has been directed at the local scene of the injury site, right? And this is kind of interesting though, because if we look at other animals, so like a planarian, for example, which is 700 million years diverged from an axolotl, we know that that's not the case. And so if we look at the axolotl, is it all just in this limb that's going to be amputated, or is there something else important going on in the rest of the body? And by important, I mean also predicated on cell proliferation. And in the planarians, it had already been shown that actually the planarians use a similar kind of almost like a two-step mechanism, and they have these little neoblast cells, which are stem cells sprinkled throughout their bodies. And what happens is that if you cut this animal in half, the head will grow a tail and the tail will grow a head. But the first thing that happens is that the neoblasts throughout the whole body get activated. So they re-enter the cell cycle everywhere. And then within a couple of days, that activation gets refined just to those amputation planes. And so we had sort of that goalpost in mind. And then in the last few years, there's been some really elegant studies to come out of Tom Rando's lab using mice. And mice are still 350 million years separated from axolotls. And the intriguing thing about mice is that they do have a systemic injury response, but they also don't regenerate limbs. In fact, if you amputate their whole limb, they can't grow it back. But if they receive a very tiny amputation of just their fingertip, then they can grow that back. And so in those studies, they showed that mice do have a systemic and local injury response. So injury of one of the muscles in the leg will activate cells even in the other leg. And in that paper, they defined a new cell state called the G-alert state, which really primes these stem cells to be quicker to respond to future injuries, which is really cool. However, as I said, the mice don't regenerate entire limbs. And so we really think about the salamander as being able to inform this from sort of an evolutionary perspective. What is happening elsewhere in the salamander body? And is it a mistake to only look at what's happening in the amputated limb? And we sort of stumbled into this question in some experiments I'll show you in a second. But before I go there, I just want to also note that mTOR signaling has been implicated in both planarians and in mice in controlling these systemic injury responses, which I'll talk about later. Okay, so in this experiment shown here, which is now published, what we found was that when you amputate a single or two limbs in a salamander, the limbs that were not amputated really can't be considered inert bystanders. And that's because if you are to actually survey who is proliferating following an amputation, and we do this by EDU injection shown here, that if you look at the regenerating limb, you do see this high induction of proliferation, which of course we would expect, especially given what I just told you what blastemas do. And so you can see it gets really cranked up in the week or so after the amputation. And so all of these hot pink cells are the proliferative cells. And here we see a really nice, robust mounded structure, which is the blastema. And in the uninjured limbs, we really see hardly any proliferation whatsoever in an animal that has no injuries. However, in the animals that have one or two limbs amputated, in the other limbs that were not amputated, we're actually super surprised to see that there's an induction of proliferation over here as well. And this is quite statistically significant, shown down here. And we do know some of the details about what some of these cell types are, but we don't actually have a whole catalog of what all the cell types are yet. And that's been quite a challenge to obtain in the axolotl. And so all I can tell you right now is a sizable fraction of these activated cells in the contralateral limbs, or the systemically activated cells, are muscle satellite cells, which are the stem cells that replenish the muscle lineage after an injury, for example, and during homeostasis. Okay, so now we know that actually following a single amputation that the animal actually activates potentially progenitor cells throughout its entire body. And we actually also saw this effect from a single limb amputation in the spinal cord, the heart, and the liver, which is super interesting. And we have a lot of questions about this. One of them is, how do these distant cells know to enter this activated state in response to an amputation? And so I'm presenting a couple of possible modes of action shown here, and these are very broad. One of them is the nervous system, and the other one is the circulatory system. And I think most people's money would have been on the circulatory system because it makes a lot of sense that there could be some molecules that are secreted that travel around the body and sort of tap these stem cells and tell them what to do. And additionally, in that mouse paper, it was also shown that this effect can be mediated by the circulatory system. However, planarians don't have a vascular system, and it's interesting to think about that when thinking about what the evolutionary implications of this are. So we actually have done a proteomic study of regenerating axolotl blood, and it's quite interesting, but I'm not going to talk to you about that today because in the meantime, we tested the nervous system, and we were really surprised to find out that what I'm going to show you next. And so on the left is just sort of the setup for the experiment. So in these experiments, what we do is first we had to prove that we could denervate these uninjured limbs and that the denervation procedure works. And so on the top, you can see on the left is an animal that has been denervated in both of the forelimbs. And so it can't really hold its arms up properly anymore. Whereas on the right is a sham operation. Basically, the animal's knocked out and it has exactly the same operation and, opening, et cetera, as the denervated one, except for don't cut the nerve. And then you can see down here, if we take these animals 3 days after the procedure and do the histology, that the denervated animals, if we look at the red staining, which labels the nerves, you can see that it's starting to disintegrate and have the spotty pattern because those distal nerves are degenerating because they've been separated from their cell bodies, and so they're dying. And then meanwhile, if we look at the sham control, you can see that the red staining, which is the nerve, is really nice and robust and intact. So we know that we can do the operation successfully. And this is how the actual experiment looks. These experiments have been largely performed by my most senior graduate student, Doigu Pezendogru, along with an awesome technician, Sarah Wilson. And what they do here is that they give both the left forelimb and left hind limb an amputation to an animal. And at the same time, in the same surgery, they go on the other side and they denervate these responding limbs over here. And then they wait a certain amount of time. In this case, I'm going to show you 14 days post-amputation. And they inject EDU and they harvest 18 hours later. So this is an extremely short pulse. And all they're really looking to see here is which cells are actually in S phase at that precise moment. It will be really interesting to look at longer pulses, but actually we haven't done that yet. Okay, so they're really asking this question, do the responding distant tissues require innervation in order to activate some cells and get them in this altered cell state of re-entering the cell cycle? And so what we found is that nerves are required for systemic response to amputation. And this is really shown here. So on the left, we have a sham operated limb tissue section. And you can see here that all the nuclei are stained with DAPI. The EDU positive cells are hot pink. So those are the cells that have re-entered the cell cycle in the sham operated animals. And then green are the nerves. And you can see that this sample has a lot of innervation. And also, if you look closely, a lot of cells that are in the activated state. Meanwhile, the responding limbs that were denervated during this experiment, you can see that two things: they're lacking innervation, which is an internal control for the experiment, but they're also lacking almost all of the EDU positive cells in the internal tissues. And this effect is quantified over here, and we've done it many times and with many different controls, and we have reproduced it. So we think this is really interesting and actually quite surprising because we had assumed probably it was going to be the circulatory system. Okay, so this has led us to sort of revise the model of what's happening after an amputation in an axolotl. So in this model, we have the left limb being amputated, and the next thing that happens is that there's this early systemic activation pathway that really makes some cells throughout the whole body enter this state of activation and tells them to synthesize new DNA and to undergo at least one cycle of cell division. We know that. And we don't know all the signals that are making this happen. But so far, we have shown that actually the nerves are required. The next thing that happens is that only some of the cells are cued to enter, to convert into blastema cells. And this only happens at the site of the injury as shown here. And so this really begs the question, what are the signals over on the amputated limb that are going to make those cells coalesce? And, for example, it would be really interesting to take a look at the collective cell behaviors over here. And you might have noticed in those early graphs from the published work that actually the effect is very significant on both the contralateral limbs as well as the amputated limbs. But the amputated limbs, it's higher. And so it's possible that these cells on the amputated limbs, are, since they're more numerous, that they're somehow signaling to one another and telling each other to come together and make that blastema. And we also do not know what resets the distantly activated tissues and tells them to stop responding. So nobody has noticed and seen an animal getting dramatically bigger everywhere else in the body, or the other limbs getting bigger. And we also know that eventually it goes away. And so we would like to know what are the signals that extinguish this systemic activation. And then finally, the amputated limb is going to regrow like we saw in the video. So far we know that nerves are required for this first step. And what we want to know is, how do you go from activated to converted? And really, what is the key difference between these two cell states? And to look at that, we've actually started making these transgenic animals, which would be really cool to collaborate with some people once they're more mature. And so in these animals, we are leveraging the Z-Fuchi technology, which has been developed in mice and also zebrafish. And basically, different stages of the cell cycle are labeled with different fluorophores. And so this would give us the opportunity to collect those cells and learn more about them, because they're still relatively rare in the tissues. And then we're also using leveraging the data that we got from that study I showed you earlier with the bulk tissue sequencing. And we're using these kinds of genes like this one here, which is called KZALD1, to generate blastema reporters, and so that we can have live animals that will tell us when we've entered that cell state. And I think this is going to be really interesting, because even though it's like extremely enriched in blastemas versus intact limbs, we actually don't know if these distantly responding cells in other limbs or elsewhere in the body might also activate these blastema reporters, but only transiently or at a milder level than the amputated limb. Okay, so I kind of gave you a hint about the fact that we might want to take a special look at this mTOR signaling, given that it had been implicated in systemic injury responses in both planarians and in mice. And so in this experiment, what we did was to give animals rapamycin, which inhibits mTOR signaling. And these are two columns of control animals, and then the rapamycin animals on the far right. And the top row here is the side that was amputated. And so what you want to see there is that basically that there are lots of nuclei, and that they're building a blastema, and that lots of proliferating cells, and that the phosphorylated S6 level, which is a readout of mTOR signaling, goes down in the rapamycin treated limbs, which indeed you do see. So the green signal is down. So that's kind of an internal control. And then I would want to draw your attention to the fact that in the contralateral limbs, so these are contralateral to amputated limbs, what we see is that in the rapamycin-treated animals, we really hardly see any in the internal tissues undergoing the cell state change to the activated re-enter the cell cycle state. And this information is quantified here. So you can see that in either of these controls, so saline or the vehicle for dissolving the rapamycin, we still see the effect in the contralateral limb to having an amputation elsewhere. But in the animals that got rapamycin, we don't see that. And so we believe that mTOR signaling is going to be important for transducing this signal once the cells receive it in these distant tissues throughout the body. Okay, so what are we doing to figure this out? And this is where hopefully we can get some more collaborators on board to take a look at this. So here we have two, cool. So here we have two schematics, and I just want to also point out that Stephen Blair is our resident bioinformaticist in the lab, and so he's on it, but we have a ton of data, and it would be great to also get more people involved. Okay, so at the top is an innervated animal, so that's going to be competent to undergo systemic activation, and here we have a denervated animal, which is not. And you can see here that they both got amputated on the other limb. This one has systemic activation and this one does not. And so what we've done is RNA-seq and differential gene expression analysis this. So we started with just bulk, and we started with micro dissections of these tissues. So you can get enough RNA out of the axons and dendrites and exclude the cell body. So basically take the RNA from the nerve right in here, and then also from the soft tissues nearby, such as the muscle. And so we've done a lot of bulk tissue RNA-seq and differential gene expression there. And we do have some handles on some of the pathways that might be operational in controlling this, but we're just now getting to the point of functionally testing them. So finally, what I want to wrap up and show you that I've told you that systemic cell cycle reentry happens in response to an amputation throughout the whole body. And now we know that nerves are required for this response. And we also know that an axolotl mTOR signaling is required for this. And we have a lot of questions still. So what are the factors that communicate these signals? And then how do the responding cells detect them? What's the full repertoire of responding cell types? This is going to be a really big question, but it's also actually quite hard to answer this question in the axolotl. So hopefully sorting some of the cells and then looking at their gene expression and classifying them will help us solve that. What happens to the responding cells? Like, how does that effect get extinguished? And do these cells just go on to live happy lives and act like normal stem cells, or are they maybe cued to die or undergo senescence or something like this? And then finally, from a broader perspective, obviously, we would love to know what is the function of systemic activation in axolotls. We have some theories, which I'd happily talk with you about later, but how does this relate to other species, including species that do regenerate and then species that don't regenerate? Here's my email address. And then finally, I just want to thank all the people and the funding that was involved. So the people who are bolded here are the main ones that have been involved in this specific project, as well as the funding, and I can take questions.
Okay, that was fantastic. I really enjoyed that talk. We have a very active chat. Let me just pull up the questions here. So I'll start right from the top, the most popular question. Swetha Murali asks, or says, really great presentation, and asks, is it the neuronal cells themselves that are necessary for regeneration, or is it the axolotl equivalent of satellite cells, Schwann cells, or some other nerve sheath associated cells?
So do you mean for needed for regeneration or for the systemic response? So like we know that both nerves and satellite cells are required for regeneration of either the full limb in the case of the nerves on the amputated side, or the muscle lineage for the regenerating limbs. So like those things are both involved in the amputated side in making it work. But we don't know on the other side yet if, for example, the nerve is somehow stimulating the satellite cells to do something, but maybe it could be bypassed. I don't know if that's really the question.
I don't know if we have a way to follow up.
Well, we might be able to so somebody like 50 years ago gave them Botox, or gave salamanders Botox, and then they showed that actually, even though they can't move because they're paralyzed, that they can still regenerate limbs. So if that holds true today, which I'll assume it does, but maybe we'll repeat it, then maybe the muscle movement per se isn't the thing that's required, but some kind of signal from the nerve.
Okay, and then that's great. And then next from, let's see, okay, from Mikhail Pepelenbosch asks, in the denervation experiments, does the nerve show a regenerative response that might complicate interpretation?
Right, so that actually does happen, and I didn't talk about this, but we have an entire PNS regeneration project in the lab that's actually in many ways, like from the gene standpoint, more advanced than this one. And so we actually have the opportunity to subtract those aspects from the data analysis and to compare them and stuff like that. So I totally appreciate that. And I think that's a really good question, because they do spontaneously regenerate peripheral nervous system way better than people do. And within 2 weeks, they can bridge a transection and the limb is totally functional again.
And actually, just following up on that, I was curious, you mentioned this briefly, but the time scale of the response. So I'm curious, the time scale of when you amputate to when you start to see something at the site, but also the time scale of the systemic activation. Like,
right.
What's faster?
Right. I think something right at the site, as far as like a reactive oxygen and these kinds of things, is probably faster. But as far as proliferation, like we detect that to be happening just as early as on the amputated side. And basically in that graph I showed, it only started at 3 days. So the earliest time points we've sampled has been 3 days. But the interesting thing is that the peak is much later, like 9 days or days or around there, which actually opens up the door to the question, like, is there some kind of central processor involved in this? I don't think that, there's like diffusible molecules and that this is the answer. I think that there's some kind of integration of the fact that the one limb has been amputated in the like a system-wide integration event.
And this next question from Wayne Stellert says, and you addressed this briefly, but it's a good question. It says, what is the consequence of the induction of proliferation in the unamputated limb? Is it balanced by apoptosis to maintain total cell numbers, or does the limb actually get bigger?
Yeah, I don't know the answer to that. It doesn't get obviously bigger, but I think this is a really cool question from like an evolutionary standpoint, because if those of you who live where a lot of salamanders live, this is the great time to go out and find them at night. And they usually reproduce in these vernal pools, which are like short-lived tiny little ponds from melted snow. And what's super cool is that they get together and they just have tons of eggs. Like each female of an axolotl will lay like 300 eggs, and some salamanders more. And so these pools will be teeming with thousands of conspecifics, so like salamanders and their brothers and sisters and cousins, but also other members of that same species. And they're incredibly cannibalistic. And that's kind of interesting to think about, because like if you leave them to their devices in the lab and house them together when they're that young, they just eat arms and legs, and they don't have any arms and legs. You have to separate them, right? We have a whole different story about that, about repeated injury, and like the fact that you can actually shut down regeneration by repeated injury. But I think from a selective advantage standpoint, like having these cells possibly poised elsewhere to more quickly respond to an injury is not a crazy idea. And so it's possible that that is one of their functions. And in the mouse system, it was shown that like they are sort of poised to more quickly recuperate or repair. And so, but without really any really great reason in a mouse. But I think in a salamander, there's actually a great reason to do that.
We have a lot of questions, and so we'll ask some people to follow up at the forum. But I want to do the next one, maybe one more before we go to break. The next popular one, which is from Joshua Thayson, who asks, "Have you done unilateral denervation to see if the response to the amputation signal is transmitted to the body by the nervous system and then transmitted to the contralateral limb humorally?"
So that's a really smart question, and we are doing that right now. So I don't know the answer to that, but we must know the answer in order to understand this system. Yes, it will be really cool either way, I think.
Great. Well, this has been a fantastic session. Thank you very much, Jessica. I appreciate the talk.